Hello everyone, this is the Theoretical Doctor and welcome back to my channel. If you want to access flashcards on this topic and other videos, do click the link below. I post weekly videos every Tuesdays and Fridays, so do subscribe for more content. So today's topic will be on carbohydrates. I will be covering monosaccharides. Alright, so just a quick introduction. Carbohydrates are organic compounds made up of carbon, hydrogen, and oxygen. So it's important to remember it's made up of carbon, hydrogen, and oxygen. So this is actually the formula for carbohydrates, and I will talk about why it's important to know later. It's divided into monosaccharides, disaccharides, and polysaccharides. So mono meaning one here, di meaning two, and depending if it's oligo or poly, it's more than two. So you can see the values over here, mono 1. If it's just 1, you add the 1 here, it will become C, H2, and O. Now if it's di, it's 2, replace it with the N value here, so it's C2, H4, O2. And oligo and poly, depending on the number that they give, you add in there, and yeah, that's what you will get. Now, monosaccharides. Monosaccharides are actually simple sugars and they contain only one basic unit. So remember, it's only one basic unit. It's the smallest carbohydrate molecule, like that's the very beginning, the very initial part, the building block for carbohydrates. They cannot be broken down further into smaller molecules by hydrolysis, like basically that, like I said earlier, it's the brick. And yeah, it's divided, they have two types. There's aldehyde and ketose. It's important to know the structure. So for aldehyde, the carbon here is connected to one atom of hydrogen and one oxygen. Whereas for ketose, it's purely oxygen, and actually on top here, it will be connected to another carbon and another carbon. So we will go into a few examples later. So it's important to note that for aldehyde, it's in, you see the double bond to the oxygen here, it's in the first carbon, and for ketose it's in the middle. Okay, so some physical properties: it's sweet, it dissolves in water, and it can be crystallized. And for chemical properties, like I mentioned, it exists as aldehydes and ketones. They are all reducing sugars. So what is reducing sugar? It means that it actually reacts with Benedict's or Fehling solution, and when it reacts with the solution, it gives a brick red precipitate. And this is due to the presence of free aldehyde. So it's just important to remember that it reacts with Benedict's solution and gives brick red precipitate. So let's go to the first one, 3Cs. So remember the formula earlier. Now if you put 3 inside the formula earlier, this is what you'll get. C3H6O3. And there are two types. There are two examples that I'll show here: glyceraldehyde and dihydroacetone. Now remember I mentioned earlier that they have aldose and ketose form. So if you see that the double bond of oxygen is on the first carbon, that's aldose. If the double bond is on the carbon that is in between, not necessarily the second, it can be, for others it can be maybe the fifth or what, but basically you see that it's connected to oxygen and the rest is connected to carbon. It doesn't connect to hydrogen, whereas here it connects to hydrogen. So that's how you differentiate. Aldose is like this, it's always on the first carbon, and for ketose, the double bond of oxygen is somewhere in between. Alright, so trioses or 3Cs, tri, are monosaccharides that contains 3 atoms of carbon. So yeah, it's very easy to remember that. They are intermediate products for metabolism in glycolysis and photosynthesis, which you will actually learn in the subsequent chapters. And some example to take note for exam: glyceraldehyde and dihydroacetone. The next one is pentoses. So pentagon, it's 5. Now again, fit it in the formula given earlier. This is what you'll get: C5H10O5. Okay, they exist as aldose and ketose. Take note of the structure again. Where is the double bond of the oxygen? It's here in the first carbon, whereas for here it's in the second one. do take note that it doesn't actually have to be fixed in the second one. It can be in the third one, it can be in the fourth one. So as long as it's in the middle, it's not the first carbon, that's a ketose. Okay, so some examples: ribulose, deoxyribose, and ribose. So the function of these pentoses, actually, they're involved in the synthesis of nucleotides. You'll learn this again in the subsequent chapters, so I won't go much into it. And ribose is used for RNA, and deoxyribose for DNA. And just remember, pentoses are monosaccharides that contains 5 atoms of carbon. Third one is hexoses. So hexagon, 6. Again, fit it in the formula here. You will get C6H12O6. And take note of the placement of where the double bond of oxygen is. If it's in the first carbon, that's an aldose. If it's somewhere in the middle, that's a ketose. Okay, so some examples: glucose, galactose, and fructose. Now, what are some of the functions of hexoses? They're used in respiration as a source of ATP. That's adenosine triphosphate. You will learn that in the subsequent chapters again. So these are just, introduction. Essentially, ATP is actually used when glucose is broken down into water and carbon dioxide. Now, for cell turgidity, it balances the water potential inside and outside the cell. It also synthesizes disaccharides, which is the next topic. Now we're in monosaccharides. And it also synthesizes polysaccharides. Like I mentioned earlier, this is the building block. It's the brick. Okay. And it also synthesizes fats and amino acids. Alright, so if you've reached this point, thank you very much. Do subscribe and share this video to those who might find this helpful. I'll see you in the next video. Bye!